Hello, everybody, and today we're going to be talking about the muscular system. This is a follow-up to our skeletal system last week. If it looks like my screen's being a little bit weird today, that's because it is being a little bit weird. I've plugged in my drawing tablet. So this is my drawing tablet. It mirrors the screen that's right in front of me, and I'm in presentation mode. And why does that look like it's muted? I don't think I'm muted right now. I'm just gonna pause. Looks like we're fine. I'm just making sure everything's not glitching out because this thing is plugged in. So it's my drawing tablet. I find that muscular physiology, muscular anatomy and function and vocabulary is quite extensive, and then the physiology that goes off of that is quite involved as well. It's very, very specific cell biology. And when people are taking typically an anatomy class, it's really the first really challenging physiology that you get. It's not impossible. It's not difficult. What it really requires is repetition. I do find that it's helpful for me to illustrate some things and for you at home to be illustrating along with me. So if you happen to have markers, colored pencils, lots and lots of things with which you can illustrate, I highly recommend getting out a couple of spare pages of notebook paper and following along with your notes today. So we're doing the muscular system. We're going to start with muscle structure in general. We'll go over the organization of muscles as we work our way down from the gross anatomy to the microscopic anatomy. And like I said, there's quite a bit of vocabulary there. That's the first thing that we're going to illustrate. And then we're going to go through the physiology of muscle contraction. Now, I think this phrase isn't even in the PowerPoint, but it's on the study guide. It's the sliding filament model of muscle contraction. It is our physiologic understanding of how we contract those muscles. So like I said, it's a very, very specific bit of physiology that has a lot of significance to you clinically. Every single detail in it is important clinically. I wouldn't be teaching it to this level in a cell biosystems overview course if it didn't build considerably. So this is going to be pretty in-depth compared to a lot of the things that we've seen so far, and absolutely necessary. It's going to give you a leg up later. I absolutely promise that. And we'll also go through some other details if you're into exercise physiology at all. There will be some details of interest to you today. And then finally, we'll cover a little bit of pathophysiology, some disorders of the muscular system. Now, the lab content is going to cover a certain list of muscles to know. It's by no means going to be a complete list of every muscle in the body. There's like 700 of them. Different sources have slightly different numbers. Fun fact, when you're dissecting a cadaver, you can just kind of peel things in two. So every once in a while, somebody discovers a new muscle or a new ligament, and I'm just like, is that really a new muscle? Or did they just take one and discover there's a good spot where you can peel it into something that looks like two muscles? And that, yeah, that's about an annual occurrence in my life. Okay, so don't worry about this list here. I will cover this in lab content. Now, what do muscles do? Of course, they are there to contract, which means they're going to pull on your bones and they are going to cause movements. They move the bones. They also, they're working all the time. I'll define muscle tone later. You have certain skeletal muscles that are always just contracting a little bit, just here and there. Things that are like postural muscles. Every time I talk about postural muscles, if we're in class in person, I watch all of my students suddenly go like this, and it's really fun to watch. It's something that I miss about in-person classes right now. So we have movement, and we have just maintenance of things like posture. We have things that work synergistically. So for example, your biceps brachii muscle causes flexion of the arm at the elbow. However, there are other muscles in the upper arm and the brachium that are also causing flexion of the arm at the elbow, and we consider those to be synergists to that biceps brachii muscle. You can do that for any muscle. So take an action. Extension of the arm at the elbow is going to be caused by, let's say, triceps brachii muscle. There are also other muscles that cause extension of the arm at the elbow. Those are synergists for the triceps brachii muscle. And then those things are antagonists to each other. So if we're looking at the biceps brachii that causes flexion of the arm at the elbow, then its antagonist would be extension of the arm at the elbow. These are really just definitions. It's not quite a muscle function so much as these muscles are working together with their common functions. So this is where we're going to start our microscopic anatomy and our organization and our understanding there. So what we're talking about is your muscles are bundles of bundles of bundles of bundles. So think of like a really thick rope. You know, if you're making a rope, you start with strings and you wrap strings together, and then that gives you a sort of thicker thread, and that you'll take several of those thicker threads and you'll weave them together, and you'll take that small rope and you'll weave it into slightly larger rope, and you'll take that and you'll weave it into a very thick, strong rope. And that's kind of the idea of what's going on with the organization of your muscles. So what you have is the muscle itself, and your muscle itself is going to be made up of muscle fascicles, and each muscle fascicle is going to be made up of several muscle fibers, and each muscle fiber is going to be made up of several myofibrils, muscle fibrils. So their prefix myo means muscle, and that means we're going to be seeing that quite a lot today. Let's make sure I'm still recording. I'm sure this is looking pretty weird at the moment. I don't know if this will let me move. Yes, okay, I've got a small area to work with. Every time I plug in my illustrating tool, my tablet, I get just a super weird view. I'm going to take my face out of screen and just illustrate this organization for you. Okay, I'll start with black, just because you're probably starting with black. Why not? I could start with red. Now let's start with black. Okay, so you have, what I'm illustrating here is sort of the big view. This is a muscle. And your muscle is made up of several bundles. You know what? I need to undo that drawing, that line right there. There we go. Let's do this. I may not be giving myself enough space right there. Okay. SLE. Now, each of these is a fascicle. This fasci right here, that actually has the same word origins as fascism. Fascism is actually based on the concept of when we are together, we are strong. So a fascicle, as an ancient term, actually means like a bundle of sticks. So it's easy to break one stick, but it's not easy to break an entire bundle of sticks. Therefore, when we act together, we are strong. Of course, that's not how fascism plays out politically, but that's actually just a shared etymology is what that's called. That word root comes from that same place. So all we're really indicating with this word fascicle is a bundle, but a bundle of what? We're talking about bundles of muscle cells. So what we have at this next level, there's a couple of different ways to phrase it. I'm going to put it as a myofiber. It's perfectly safe to say muscle fiber or just fiber. The thing about this is that it is a cell. This is the muscle cell, and that's actually kind of important. So myofiber, we are now discussing a very, very long but very, very microscopic cell. So it's still something that's only going to be visible in histology with a light microscope in terms of its thickness, like this diameter right here. That having been said, these myofibers can run the entire length of a muscle, so they can be quite long. Now, within the plasma membrane of this highly specialized muscle cell, you still have bundling. We have more anatomy here. And what I'm going to go ahead and call this is a myofibril. And it is made out of some very important pieces of cytoskeleton called myofilaments. This is why I stuck with that word myo right there. Up here, it gets a little confusing when everything starts with myo, but I want to make sure that you're separating those suffixes. Fiber, fibril, filament. That's what's important. So myofilaments are pieces of cytoskeleton. And that's just going to have a lot of really important function here. It's just going to have a lot of importance and significance to how our muscles are actually going to contract. Now I'm going to make this a little bit more complicated for you. Remember how, and it's not me making it complicated, I'm just pointing out something that is. Remember how skeletal muscle is striated in appearance. It's stripy, right? So you have all these stripes, and one stripe from beginning to end is known as a sarcomere. Now, I have really limited space. This is shrunk down really, really small from my view. I hope you have a better view than I have. So I know all of these words are cramming in together. Feel free to illustrate this in a more stretched out way. Now, a sarcomere is the contractile unit. And what I mean by that is that the cytoskeleton in this sarcomere and the way that it is engaging together, arrow, this is how contraction is actually going to work. This is the level that contraction is occurring on. It's occurring in the sarcomeres. These sarcomeres are repeating all along the length of these myofibrils, and all of these myofibrils are repeating along the entire length of the myofiber. And of course, all of these myofibers are bundled into fascicles, and all these fascicles are bundled into the muscle itself. And I'm just going to point out these connective tissue sheaths because you do have them labeled in some of your images. You do have several layers of connective tissue. So around the entire muscle, you have something called the epimysium. Let me change colors here. I'm going to purple. Around each muscle fascicle, you have something called perimysium. So again, that means around this fascicle, and there's another fascicle, and there's another fascicle, and there's just connective tissue in between these spaces, and it's actually helping to distribute things like blood vessels and nerves when we're talking about perimysium. So there'll be little arteries and veins in here, and nerves we almost draw in a sort of a orange or like creamy color. So that's what that perimysium is about. And then immediately surrounding each myofiber, there's also a connective tissue layer known as the endomysium. It's still one word, by the way. That's an I, not an E. It's still one word, endomysium. I just don't have a lot of space here. Now, what I find is that when I am lecturing this in person, you absolutely want me to repeat this about 500 million times. I'm going to repeat it one more time. I'm just going to be really, really clear and succinct with it. And then you can, this being a YouTube video, you can rewind, replay. So go back to about 1330, where this is when I'm starting this. So the muscle is surrounded by epimysium. It is made up of muscle fascicles surrounded by perimysium. The fascicle is made up of myofibers, which are surrounded by endomysium. The myofiber is segmented into myofibrils. The myofibrils contain myofilaments, which are pieces of cytoskeleton. Along the length of the myofibril is the contractile unit known as the sarcomere. Now, once in the reverse order, the sarcomere is the contractile unit of the muscle. They extend along the length of the myofibril, which is made up of several myofilaments, which are a type of organized cytoskeleton. The myofibril are just segments of the muscle fiber known as the myofiber. The myofiber is surrounded by endomysium. The myofibers are bundled in several together into something known as a fascicle, which is itself surrounded by perimysium. That perimysium is going to distribute nerves, arteries, and veins. Multiple muscle fascicles will bundle together into a skeletal muscle, which will itself be surrounded by a connective tissue sheath known as the epimysium. Okay, so that's the end. I highly recommend going back to 1330 and repeating that about 1 billion times until you know it. Okay, so that's all reflected on this as well. Now, it's always in your textbook. You can always have a nice medical quality illustration of this in your textbook and your PowerPoint. But like I said, I do find it's more useful to draw it out yourself if you actually want to understand this organization. If I was to say how long this usually takes in a lecture, if I actually have time for it, I would say I usually spend about half an hour reviewing that. So I actually recommend pausing it, spending a good half an hour until you feel really comfortable with that terminology. If you don't feel really comfortable with that terminology, this next bit is going to get that much harder. And that's true for every segment of this lecture. So every time I bombard you with a bunch of new vocab, I recommend spending more time on that vocab until you understand it before moving on to the next segment. Now, I do want to point out a couple of things here. Is your cellular anatomy in the muscle is really specialized. And because it's so specialized, we actually have some different naming conventions. So depending on which resources you're using for your studies, you may end up coming across words like sarcoplasmic reticulum, sarcolemma. What's the other one? There's another one too. But that word sarco, that prefix sarco, it literally means flesh. And anytime you see that word flesh, frequently there's a connection to muscle tissue. Not always, but most of the time. So instead of this being called the plasma membrane here, we call it the sarcolemma. It's the same thing as the plasma membrane on any given cell. It's just highly specialized in this muscle cell. It's highly reinforced with a lot of really important proteins that anchor the cytoskeleton to it, make it really strong. And then instead of an endoplasmic reticulum, this sort of membranous organelle that in other cells would be called the endoplasmic reticulum and have those metabolic functions, here, instead, it's going to have a storage function, and because it's so specialized, we're going to call it sarcoplasmic reticulum. Let's see. Now, this is our stem cell lineage, just real quick. You go from having myoblasts, so again, the blast is an immature form of the cell, and then they become immature muscle fibers, and then they become the adult muscle cells. Now, something that is actually kind of clinically significant here is that these muscle cells are non-mitotic, especially in adults. This is non-mitotic tissue. These cells do not undergo mitosis once you have developed your sort of base set of muscles. Once you've grown to a certain extent, they stop undergoing mitosis. That has clinical relevance in the sense that if you damage skeletal muscle or cardiac muscle for that matter, or smooth muscle, it being non-mitotic, you can't replace those cells. That injury is going to heal with connective tissue, which is not going to be contractile, it's not going to be excitable, it's not going to be able to shorten, it's not going to be able to do its job. So once you've damaged one of these pieces of muscle, it's very difficult to get that function, that same level of function that you had before an injury. Some more vocab. We are stacking on the vocab today. It's just how it's going to work. The arrangement of your myofibrils is that they are going to be surrounded by some specialized membranous structures. You're going to have a sort of tube, and that tube is just an extension of the sarcolemma, which I'll remind you is the plasma membrane of this highly specialized cell. So imagine in your highly specialized muscle cell, a tube made out of plasma membrane, a tube made out of sarcolemma. We call that a T-tubule, and its job is actually going to be to, I'm just going to say the word propagate action potentials. You'll understand that a little bit more later. So, go ahead and fill that out in your notes. T-tubule, propagate action potentials. At the end of the day, you can come back to that idea and fill in your understanding of what an action potential is. We will find adjacent to those T-tubules the ends of the sarcoplasmic reticulum. And it looks like we're just sticking with the phrase sarcoplasmic reticulum. There is a specialized name just for this end here. It's called the terminal cisternae. But it looks like we're not dealing with that just now. So we have the sarcoplasmic reticulum butted up to this T-tubule right here. So this electrical impulse, this action potential propagates down the T-tubule. That's going to cause a change in the sarcoplasmic reticulum. The sarcoplasmic reticulum is going to store calcium. So again, right now we're just working on definitions. So in your notes, you're going to want to write sarcoplasmic reticulum stores calcium, right? And they're adjacent to T-tubules. And then we're going to put the process in after. Let's go back to this picture really quick. Now, in order to see the next part, you kind of have to take all the membranous structures away and get to the cytoskeleton. So here's the cytoskeleton down here. If you compare this and this, you can see that all of the T-tubules and sarcoplasmic reticulum are simply around each myofibril, right? They're surrounding it. So we take those away so that we can see the myofibril, and then we can see the organization of the cytoskeleton, and we can start looking at those sarcomeres. So, what's a sarcomere? The sarcomere, like I said before, is the contractile unit of the muscle cell. So the thing that actually shortens is this one unit. We have something called Z-line, Z-line to Z-line. All of the cytoskeleton within that unit, that's the sarcomere. So what my hands are illustrating right now, let's have this knuckle be a Z-line and this way. This knuckle be a Z-line, this knuckle's a Z-line. There you have one sarcomere and one unit of muscle contraction. The cytoskeleton here has a very, very specific arrangement, and as such we have some really, really specific names for the proteins in that cytoskeleton arrangement. There's two types of cytoskeleton here. They are known as actin and myosin. Myosin is thick, actin is thin. I used to make my students listen to a whole rap about it, and that was the only thing they'd ever get out of this rap, and it's one use. Myosin is thick, actin is thin. So you have these two different sizes of cytoskeleton, and they make up the sarcomere. So you're going to have your myosin here, you're going to have your actin here, and you have some proteins anchoring just at the ends. Those make the Z disc. I've been saying Z line. It's fine. They're both accepted. And that is one sarcomere. So that's how they relate, is your sarcomere is made up of your cytoskeleton, which is your actin and your myosin. And the Z line is defining the edges of one sarcomere. So again, one sarcomere is one from Z line to Z line. I know the vocab is crazy right now. It's a lot. But wait, there's more. The good thing about YouTube is you can rewind, you can listen to it again. One thing I'm going to do is I'm going to give you some outside resources. So the way we treat this in person is I teach it to you. I give you all the vocab. I go through muscle contraction once in class. And then we watch a McGraw-Hill video about the topic. And then we watch a crash course video about the topic. And it's just going to be a bunch of different voices giving you the same information in repetition. So usually by about the third video that you've watched on the same topic, that's when you start to understand it. That's when it starts to click. So if it's not clicking right now, that's really, really normal. So just keep on filling out your vocab list right now. Keep on filling out what you can in your study guide and just listing things and defining things and having a point of reference for all of these words to look at later, and let it wash over you for now, and then come back to it when you have greater understanding. So again, sarcomere is from Z disc to Z disc. Within the sarcomere, you have actin and myosin. Myosin is thick, actin is thin. We're going to have a connection between actin and myosin. We're going to have something called a cross bridge, where myosin heads really want to bind with binding sites on the actin. So this is the cross bridge right here. But something is blocking the myosin head from binding to the binding site on the actin. And that is a protein known as tropomyosin. Tropomyosin is a string-like protein that, in its passive state, blocks binding sites for myosin heads on actin. So it's wrapping around the actin. Yeah? There's another protein here, and its name is troponin. Some people pronounce it troponin. I always kind of illustrate it like a little snowman. I kind of ignore this bit and just like draw two little guys, and that's my troponin. There are different types of troponin, by the way. If you are already an EMT or working in some kind of emergency department, you may know that people who have, if you have a suspicion that somebody's had a myocardial infarction, which is a heart attack, you test their blood for something called troponin, and that is related. So I hope you're having that light bulb moment right there. Oh, that's why they test for it. We'll circle back around to that in just a second. So here's a protein known as troponin, and it is closely associated with tropomyosin. Troponin has a binding site for calcium. And there's a couple of rules. This is something that the crash course video does quite well, is explaining the rule of proteins. When things bind to proteins, proteins change shape. Yeah, that's one of the rules of proteins. So when calcium binds to troponin, tropomyosin is going to change shape. Okay, here's the structure of myosin. There's actually more than one kind of myosin. There's a free kind of myosin that kind of walks along T-tubules and transports vesicles in the cell. I'm getting a little bit overboard, but if you've ever watched Inner Life of the Cell 3D animation, you can see a little myosin head. They just kind of walk like this. But this is the myosin head. So what you're seeing right now, what's being illustrated, is a piece of this myosin protein that can move. This myosin head can move. So hopefully my fingers right now are representing this myosin head right here. So each of my fingers is one of these. And that's what they do, is they walk. They walk like that. But the body of this myosin head is bound up into the larger myosin filament. So while if this were a free myosin, it could walk along something. But this is a bound up myosin, so when it moves, it doesn't move the protein, it moves something outside of the protein. And that's what we're going to see. Now, our goal here is to get the sarcomere to shorten. That's what contraction is. It's a shortening of the sarcomere. So what we're going to do is pull the ends of the sarcomere towards the midline. So we have, this is kind of a histology thing. Remember the striations in our histology of skeletal muscle fiber? We actually have specific letters for each of those like light and dark bands that we can see and appreciate histologically. The most important ones, in my opinion, are the Z-disc, which we've already covered. So there's a specific protein there linking just the actin. This is the actin bound to that Z-disc along the periphery of your sarcomere. Here's a sarcomere, Z-disc, Z-disc. The myosin is bound along the M line. So there's that word M line. So there's a different set of proteins binding the myosin filaments, the thick filaments, right at that midline. M, mid, it works out, right? So these are all bound. These are all bound. The only connection between myosin and actin is the myosin heads. They're not always in contact because of that tropomyosin covering everything. Now the H zone is the sort of paler region. So you have, again, this overlap of actin, myosin, actin, myosin, actin, myosin. Where there's just actin, it's going to be a little bit paler. Where it's just myosin, it's going to be a little bit paler. The central pale area is the H zone. So when you have contraction of a sarcomere, see how even with my hands like this, you see how that works? It looks pale, and then it's very, very dense. And then it's not dense, and then it's very dense. So that density is giving us that color change. So when Z-disc gets closer to Z-disc, you narrow the H zone, and Z-discs are moving closer to the M line. That whole phrase. I'll say it one more time, just for funsies. During muscle contraction, the H zone narrows, Z disc gets closer to Z disc, which both get closer to the M line. So that H zone narrows during muscle contraction. Now again, we're looking at a myofibril along its length, and we'll have a repeating pattern of sarcomeres. Now again, this individual unit is the individual unit of contraction, but there's something called the all or nothing principle, which means that every sarcomere along the length of this muscle fiber, or myofibril I should say, is going to contract at the same time. Once one segment of a cell is contracting, every other segment of that specific cell is also going to be contracting. So again, Z disc, C disc, M line, H zone is here to here, and that does shrink during muscle contraction. Here it is if you're looking at certain forms of histology. Z line, Z disc. H zone, it looks a little bit paler, except for where you have that protein anchoring the myosin. So we're still just setting things up. I haven't gone through the whole process just yet. We're still making sure you understand every single piece of this. What we're aiming to do is have the myosin heads bind to binding sites on the actin. And they're going to, now my entire fist is going to be the myosin head. Instead of using my fingers, I'm going to have my fist be a myosin head. We want the myosin head to come in contact with the actin. We want the myosin head to bend, pulling on the actin. If you saw that, that went bind, bend, pulls on the actin, moves the actin, and then it's going to release, it's going to reset, and it's going to bind again, and it's going to pull again. So this is what the sliding filament is going to look like. So notice this arm stays stationary. This represents the myosin, this represents the myosin head. This arm represents the actin. In fact, how about I do a little thing here? There we go. Ready? There's the actin moving. Myosin head bends, but the actin gets pulled. And this is how we're going to shorten that sarcomere. So it is important to recognize that the myosin piece of cytoskeleton, the myosin filament, is going to stay stationary. It's going to pull on the actin. It's going to pull it towards that midline. One more time. Students usually write like that. Okay. Now again, that relies on the myosin forming a cross bridge with the actin. It's being prevented from doing that when the muscle is relaxed. It's being prevented from the myofilaments blocking the myosin head binding sites. Okay, 3340 is about where we're going to start my description of the sliding filament model of muscle contraction. Here's the whole story. You have a neuron, and the end of that neuron is called a synaptic knob, and that synaptic knob is going to synapse onto the neuromuscular junction, onto the sarcolemma, a specific spot on the sarcolemma. That neuron is going to release a neurotransmitter called acetylcholine. You can actually just write ACH, capital A, capital C, lowercase h, for acetylcholine. Acetylcholine is going to bind to receptors for acetylcholine located on the, what's called the motor end plate of the sarcolemma. So basically you've got a synapse, and we have receptors for that acetylcholine on the other side of that synapse on the muscle cell itself. This is going to cause depolarization down the T-tubules. An electrical impulse is going to be transmitted along the T-tubule. The electrical impulse traveling along the T-tubule is going to release calcium from the sarcoplasmic reticulum. That calcium is going to diffuse into the, sarcoplasm. There's the third word I was looking for. Instead of cytoplasm, it's so specific, we're going to call it sarcoplasm. Calcium released from the sarcoplasmic reticulum into the general fluid of the cell, sarcoplasm, binds to troponin. That's the snowman shape. But when ions bind to proteins change shape. So the calcium binding to the troponin causes the tropomyosin to shift, revealing myosin binding sites on actin. Now that those myosin head binding sites are revealed, a cross bridge will form as long as we've got ATP around. If we've got ATP, then the cross bridge can form, and we're going to have a repeating cycle of, it's going to be cocked, it's going to bind, it's going to bend, it's going to detach. Attach, pivot, detach, reset. Attach, pivot, detach, reset. The actin filament gets pulled towards the center of the sarcomere, shortening the H zone, bringing the Z lines closer together. Okay, that's the thing. I'm going to illustrate it. I'm going to repeat it a little bit. I'm not going to repeat it too much because you can always rewind. Here's what it looks like when calcium binds to troponin, moving myosin head binding sites, or revealing myosin head binding sites. Here's that neuromuscular junction I was talking about. This is the spinal cord. We have motor neurons, and that motor neuron splits, and it's going to innervate multiple skeletal muscle fibers, which again is the cell. I'm going to move my face for a second. Neuromuscular junction is the connection between the axon terminal, is the phrase I like, and the actual cell surface, the sarcolemma. Here's the motor neuron. They went with synaptic bulb, that's fine. This motor neuron is going to release acetylcholine into the synaptic cleft. This folded piece of sarcolemma has receptors for the acetylcholine. There's the receptors for the acetylcholine. They're going to let sodium in. Sodium's going to be kind of important to us. Once sodium is in, that's going to allow the cell to do what we call depolarize, which just means become more positive. Before sodium was in, outside of the cell it was very positive. Inside of the cell was very negative. Then acetylcholine bound and allowed a lot of sodium in, and sodium is Na+. It is a positive ion. So when we let a lot of sodium into the cell became very positive. When the cell became positive after having been negative, we call that depolarization. It went from being very negative to being very positive. So that was depolarization. After this happens, after the muscle contraction, the stimulus is over, we're going to go back to being very negative. And the process of going from very, very positive to very negative again, we're going to refer to as repolarization. So that's what's going to occur when muscle contraction stops, when we're done with that muscle contraction. We will talk about action potentials more during our neuro unit. I love the action potential. It is one of my favorites. And I'm very good at teaching it as well. We're going to just stick with this positive, negative. Sodium comes in, it's positive. Sodium leaves, it's negative again. That's all we'll do right now, and that's plenty. But if you're thinking, hey, you told me to write down action potential related to the T-tubule earlier and I still don't understand it, you can wait until next week and then see if you understand action potentials after next week. And then either say, yes, I understand action potentials, or no, Reeves, I need office hours. Please help me with action potentials. And we'll make that happen. Okie dokie. This is actually backed up a little bit. There's also action potentials in neurons, as I just indicated by this concept of nervous system is next week, and we'll be covering action potentials then. So there's action potentials in the neuron. They arrive at the synaptic knob. They're going to initiate a process that involves calcium. Calcium is going to cause the release of the neurotransmitter acetylcholine in this synaptic knob. Acetylcholine is going to go into the synaptic cleft, and it's going to bind to acetylcholine receptors, allowing sodium in, therefore depolarizing the membrane. So we backed up and we just reviewed that a little bit. Action potentials propagate along the T-tubules, and those action potentials trigger calcium to leave the storage sacs of the sarcoplasmic reticulum. There's a different word for that. It's a better word. Terminal cisternae of the sarcoplasmic reticulum is a much more complicated but more accurate word than calcium storage sacs of the sarcoplasmic reticulum. Whew, okay, I just fatigued for a second, so I had to pause and just catch my breath for a second, because I know I get less effective when I'm fatigued, so it's probably best to just take a little breather there. Okay, so again, action potentials propagate down the T-tubule and cause the release of calcium from the sarcoplasmic reticulum. The calcium is released from the sarcoplasmic reticulum into the cytosol in general. Now that calcium is in the cytosol, it's going to bind to troponin, moving tropomyosin away from myosin head binding sites. Now that myosin heads have access to myosin head binding sites on actin, they go through the cycle of attach, pivot, detach, reset, pulling the actin closer to the M line and moving the Z discs closer together, shortening the H zone. Now, I do want to take just a second and point out calcium was important twice. Calcium was important for releasing neurotransmitters, and calcium was important for binding troponin and allowing for cross bridges to form. So you may remember last week during skeletal system when I was saying, you know, there are a lot of physiological processes that calcium is needed for. Keeping your bones strong is not always the highest priority. If you guys remember that, now we can kind of piece together the other part of this. What if you're running a marathon and you have to power your muscles? You have to have motor neurons firing acetylcholine into that neuromuscular junction, and you have to have the ability to bind myosin heads to myosin head binding sites. Those both require calcium. So you might leach calcium from your bones during a marathon in order to power your muscles, your nerves, everything else that requires calcium. So what's more important, your brain and your muscles working or your bones being strong in that scenario? So think about how your body, your homeostasis is going to work to keep everything functioning in the short term and in the long term. Now, at some point, this process ends, right? So the nerve activation ends. We stop releasing acetylcholine into the neuromuscular junction. We stop binding acetylcholine to acetylcholine receptors. We actually have calcium pumps that return that calcium to the sarcoplasmic reticulum. The calcium is removed from the troponin. The myosin binding sites are covered by tropomyosin again. If there's no calcium around, there's no binding, there's no cross bridge. So we'll end up having... I hope you enjoyed that sound effect. And that's the sliding filament model of muscle contraction. These are the sliding filaments, and that's the model. It starts all the way up from that neuron releasing acetylcholine into the synaptic cleft, and it ends with attach, pivot, detach, reset, and then the resequestering of calcium and the return to the original length of that muscle. There's another molecule involved that I want you to be very much aware of, is this would not happen without our good friend adenosine triphosphate, our energy currency of the cell. Mitochondria is the powerhouse of the cell, right? All my internet people are going, yeah, it is. Okay, so you have your myosin head, and it's going to utilize the energy from that ATP molecule before it binds. So we say it's cocked, I know. It's cocked and ready to go when it's storing a DP in inorganic phosphate. That's what it's in the form of when it actually binds. We're actually going to get a new ATP molecule in order to detach. And that is actually important. It looks like this teeny tiny little detail, but it's actually important, I promise you. Why is it important? Well, think about a scenario in which you're ATP depleted. You've run out of ATP. Maybe you're hypoxic, you're low on oxygen or nutrients, something that you need to make ATP. Or maybe you're dead. So what gets interrupted in this process? Is it the attaching or the detaching if you run out of ATP? The part that gets interrupted is the detaching. So there may be some scenarios that you can think about where, well, that cross bridge is stuck. There's no new ATP, so it can't release, so it's stuck, and you can no longer release the muscle contraction. I'm not talking about tetanus. This is about absence of oxygen and nutrients. So I'll give you like 5 seconds to think about it for yourself. Pause it if you want more time. Rigor mortis is what I'm talking about. So when we die, we stop breathing air, we stop getting nutrition, and our muscles for a short period of time are going to be stuck in whatever position they were in when we died, and that's called rigor mortis. And it's only going to end when, decay sets in, basically, when the electronegativities are so interrupted that things just fall apart anyway. So literally when things are falling apart in the cell is when rigor mortis ends. We've got that again. We can see the narrowing H zone between these two slides. I always like to make these like a little animation right here. Wide H zone, narrow H zone, wide H zone, narrow H zone, wide H zone, narrow H zone. Okay, there it is even narrower. Ooh, I could have gone further with that. Yeah. What a great animation. Okay, so that's muscle contraction. Went too far there. Yeah, okay. I hope you enjoyed my animation there. I had fun with it. This is a tough lecture, as you might imagine. I know your brain is probably melting at home, too. You can pause, and you can come back to it when you're ready, right? So just take a week with this one. There's that again. We're in ADP when we attach. We do our power stroke. We get a new ATP to detach. And then we've already had the lysis of that ATP into ADP and phosphate, and we're ready to go again. So that means you need 2 molecules for muscle contraction. You need ATP for muscle contraction, and you need calcium for muscle contraction. ATP can be replenished via cellular respiration, which hopefully you know something about at this point. And there's also actually an alternate route. It's creatine phosphate pathway. And I actually really do like this picture. This is the picture that was here before. It's a lovely picture. It shows you that you can phosphorylate creatine really easily as a storage form for phosphate, and then we can donate that phosphate to adenosine diphosphate to turn it into ATP. But if you're really opposed to chemistry, I've got a little simple animation for you. Let me take just a second. Here is how we make ATP from creatine phosphate. That was the whole thing. It's a little oversimplified. I apologize to any chemists in the room who hated that. I had a picture of like this in one of my old lectures and I just can't find it. So I just made a little art thing for you out of shapes in PowerPoint. That is how creatine phosphate is going to get you more ATP. So it's actually just one pathway. So your body is making its own creatine phosphate. And so then athletes were like, maybe we can supplement creatine phosphate and we'll just get like super strong because we'll have so much ATP in our muscles. And I think it's interesting this is in the past tense. So obviously this slide was made by my predecessor, and I'm keeping it because she did a great job and just to be consistent with the curriculum at Northwestern in general. So past tense tried creatine supplementation. You could still go to the supplement store, to the vitamin store, and get creatine, creatinine supplements. That's absolutely still a thing. There's a good reason to learn about this. If you are testing to see if somebody's kidneys are working, one of the tests might be a creatinine test, which is a metabolic byproduct of creatine. But if you're an athlete and you've been supplementing with creatine phosphate supplementation, you might mess up on that test and it might come back with like, whoa, your kidneys are failing, buddy. You're like, no, I just, I was lifting weights yesterday and I supplemented creatine phosphate. So apparently what was found out after a bunch of athletes tried creatine supplementation is that, yeah, they got some muscle mass out of it, but their own body's creatine phosphate synthesis pathway was interrupted and they stopped making their own. So that sounds like a bad game all around. Let's not do that. Okay, so I think I'm actually going to stop here. It's kind of a weird point. I should have probably stopped before creatine phosphate. The whole first part was all the same thing. It's really hard to subdivide these images or these videos.